our voice in testing times. So, first of all, I'd say thank you very much for inviting me to come and give this lecture. I'm really incredibly honoured, both for myself and for Lazarus, to be invited. In fact, when the email arrived, I thought it must have been meant for somebody else. So it took a bit of delay to actually say that it was meant for me. And I'm also quite nervous about following in so many eminent footsteps giving this particular lecture. I have to say also thanks to the SNP for enabling me to travel in a little more comfort than I might have otherwise. So I'm really looking forward to that on the way back. One stage I didn't quite think I'd make it here for various reasons, and then once I got all the clearances to get here, the Department of Homeland Security, in the US decided I was some sort of terrorist threat, but you'll be pleased to know that after they swabbed me, I'm completely clear of explosives, so I'm quite safe to be here. Okay, now I'll just get started on my talk. Okay, just as Steve said, I trained first as a biochemist and did my PhD in biochemistry, which took, I think, 7 years, something like that. And then I decided that after all that I'd really like to be a vet, so I went off and trained as a vet as well. So in total it took me 12 years to get a qualification. And I did work for a year as a vet, but the bit of being a vet that I really enjoyed was diagnosis rather than actually treatment. So I thought, well, that sounds like a scientist, so I went back to being a scientist. I've been at Lazarus since 1988, which makes me the second longest serving staff member there. I actually worked for a while with Mary Dempsey, who I believe used to work. I think she retired in PLC and went to New Zealand to do work in her retirement, and she was still actually working at Lazarus when I was there, so there's a sort of continuity there. My expertise obviously is the on-farm hide and skin quality, which is what I'm going to be talking about. I also do all the microscopy at Lazarus, and much of my work these days is, rather than research, is actually diagnosis of defects and damage that is on things that are sent in by our members, what we call works problems. So I spend, I think, about 70% of my time on that these days. And leather type validation, which is just proving that your leather couch is actually a real leather couch. I'm surprised how many are not real leather couches. I'll just say a little bit about Lazarus. Currently, or for quite some time now, Australasia's, that's Australia and New Zealand's only hide, skin and leather research facility. It's been around since 1928, which in terms of English terms is really, really young, but in New Zealand terms it makes it the oldest research association in the country. And it's been in Palmerston North since 1973. Prior to that it was in Wellington. Actually started out in the Prime Minister's stables, and, but we now have a purpose-built building which is no doubt somewhat better than the stables. Just showing you a picture of our building and where exactly in New Zealand it is. I had to put the flag on there because there's been a referendum recently in New Zealand about whether we should get a new flag, because the argument was that it's too close to Australia's flag, nobody knows what it looks like, so I thought, well, I'll show it, and then at least there'll be about 80 people who know what a New Zealand flag looks like. It's on the outskirts of the city, which is it's the main city in the Manawatu. It has a population, I think, of 70,000, so again, by UK standards, it's a very small city, but by our standards, it's probably about the 6th largest, I think, in the country. And I chose this photograph because basically it shows what Manawatu is all about. Sheep farming, you can see sheep there, no cattle. Hills, we actually did some of our research on the far side of this range of hills. And the most important thing in that photograph is actually the wind turbines. It tells you all you need to know about Palmerston North. It's very windy. Our turbines are probably the most efficient in the world at generating electricity because it just never stops blowing wind across the river. What Lazarus does, our major function, of course, is research, but we also have quality control services. We have a physical testing laboratory and a chemical testing laboratory for members. We do fault diagnosis, that's my role. We act as consultants for the industry in New Zealand. We have educational services, running courses for people working in tanneries and fellmongeries, and also we run block courses and short courses for people who just want to learn about leather. And it used to be called a library, but now I'm told it's called information services. And our research is obviously, again, improving the quality of hides and skins. That's been my role basically for the last 28 years. We do fundamental research into hide and skin composition and structure. Some fairly cutting-edge stuff we're doing at present. And from this, we can target improvements in processing and product right through from preservation to the finished leather. So it all ties together, the various facets that we do. I've been involved in quite a number of areas of research, not just Rawstock. I was actually employed as a chemist by Tony Passman, who some of you may remember. I don't think he realised there was a difference between being a chemist and a biochemist, and I certainly wasn't about to tell him. I think he may have worked it out eventually because he actually employed a chemist about a year after I joined, obviously realising I wasn't a proper one. I've been a fellmongery researcher, we now have a dedicated fellmongery researcher, but when I started I was the fellmongery researcher as well as the Rawstock researcher. But I'm only going to talk about Rawstock research today. This is the research platforms from our industry strategy, research and development strategy. And platform 1 is improving raw material quality, and there we've got platform 2, that's the fundamental, and 3, the processing, and the other 2 are basically spin-offs from those platforms. It's our philosophy at LASRA that if you want good leather, improving processing is important, but improving the quality of the raw stock is more important, because it's a lot easier to start with something good rather than starting with something that's rubbish and trying to hide the faults and process it to improve it. And I think this was brought home to me yesterday. We actually were lucky enough to visit the Edward Green footwear shoe factory and see how selective they were about the leather they used, and how any tiny fault was enough basically to cause their product to be rejected. I mean, they showed us a shoe at the end which was one of their rejects, and after I'd been looking at it for a good while, I did see the 2 quarter-inch dents in the surface of one of the panels that were enough to cause it to become a reject. But it really brought home to me how important raw material quality is. With the process they were using, we couldn't hide faults. If they were there, they were visible, so you had to start with good leather. Just say a little bit about sheep and beef farming in New Zealand, because it's probably different to, definitely different to what in the UK and where most of you come from. The average farm size is 371 hectares, but that includes everything from lifestyle blocks of 5 hectares right up to the very biggest. So they can go up to 30,000 hectares or more, and can have 60,000 sheep on them and maybe 5,000 cattle. Just the week before I flew over here, I was reading an article about shearing on one of the stations in Southland, in the South Island. It's 14,000 hectares, but they had 12 shearers who'd been working there for 6 weeks because they had 90,000 sheep to shear, and that's their flock, their ewes plus their lambs, 90,000 sheep. So you can see the scale is very big, and that does mean have implications for the quality of product. You don't get individual attention, obviously, if you've got 90,000 sheep on your property. Land that varies from flat to what we call rolling, which anybody else would call hills, to actually very steep. It's important that beef cattle and sheep are never housed in winter, so they're outdoors all year round, which has advantages and disadvantages, of course. There are very few feedlots. I think there might be 1 or 2 in the South Island, and that's about it. And there are no subsidies for farmers, which is probably another important thing. So what they earn is basically what the product is worth on the international market. Because of course, with a population of 4 million and rather a lot of sheep and cattle, we eat very, very little of what New Zealand produces. We export just about all of it. That's the flat land. This has got sheep on it, but increasingly that's been taken over by dairy, and the price of milk is used to be very, very high, so of course every bit of land that could be converted to a dairy farm was converted to a dairy farm, so flat land is at a premium for sheep farming now. Rolling hills, even they tend to get taken over for dairy support, so there's less of that sort of land available. You can't grow dairy cattle on tussock. If you notice there, the farmer, the sheepdog and the sheep all have longer legs on the left-hand side, so they don't fall down the hill. So that's really only suitable for sheep and also for cattle. And this is about as good as it gets if you're a cattle beast in New Zealand and it's snowing, you get some hay thrown on top of the snow. But on the other hand, that's as bad as the snow ever gets as well, so it's not very deep and will thaw very quickly, so it's really quite a temperate environment even in the South Island, but they're not disadvantaged by being kept outdoors. Just to show you what the breeds are, mostly Romneys. Perrendales and Kookalas are actually Romney crosses, so Romney is the basic sheep in New Zealand. Very few fine wolves. You see there the merino section is very small, and the others would be down breeds, things like that, used mostly as sire breeds. In our cattle, again, mostly what we call the British breeds, the Angus and the Hereford, and also quite a lot of Friesians, because with the dairy industry you've got to do something with the calves, and a lot of the bull calves are reared for bull beef, so Friesian bull beef hides are quite a large part of our hide intake. And that's the dairy herd. Of course, all the dairy cows eventually get slaughtered and the hides have to go somewhere. So they're mostly either Kiwi cross, which is just a Holstein Friesian Jersey cross, or purebred Friesians, or a few Jerseys. I'm not quite certain what the others are, I think, because there are very few other dairy breeds in New Zealand, so I think those are possibly where the farmer forgot to fill in a bit of a form-set breed. I just wanted to show you this, what's been happening over since long before I joined Glasgow actually, but note the different scales there. The cattle goes up to 7 million, the sheep 70 million. So if you look at the sheep line there, the green line, at one stage we had 70 million sheep, and that's ewes in New Zealand. And it's now down to just under 30 million, so there's been a huge change in farming practices over that time. When it was at its peak of 70 million, we actually had subsidies on meat and various things, and there was actually something called a sheep retention subsidy, which meant you got $1 for every sheep you had. So I'm not quite certain how many of those 70 million sheep were virtual sheep rather than real sheep, because with farms as large as we have and the sort of terrain that we have, no government inspector was going to go and count. If you said you had 10,000 ewes, they accepted that you had 10,000 ewes and gave you $10,000. So I think a few of them might have been virtual sheep, but even so, there's been a very steep decline over that time. And the other notable thing about this graph is the blue line, the increase in the number of dairy cattle, which of course has implications for hide quality, because a lot more of the hides that are coming through are now dairy hides rather than beef cattle hides. And if you look right at the end of that dairy line, it's gone down. That's because the price of milk has dropped very, very dramatically, to about a third of what it used to be. And so a lot of dairy cattle are actually being sold off. And that again has had implications, because they've been sold to the works, so there's been a lot more dairy hides appearing in the processing. Another thing I wanted to say, and I don't have a slide on it, is there's very little trading of either animals, little trading of animals in New Zealand, and no trading of hides and skins. So for hides and skins, they're either processed by the company which did the slaughtering for meat, if they have their own processing plant, but they're processed by them, or they will be processed under contract by a dedicated processor, or they will be sold under contract. So there's no trading, there's no spot market for skins, but they're all, they know where they're going basically, so nobody's trading for them, there's no price or war to get hides and skins. You just, they go where they go, and that's it. Okay, now to the Rawstock research. We look at defects, both the inherent, which is the ones born in the skin, and the acquired ones. We looked at breed differences, we looked at farming practices, and currently we're just moving into the genetics of skin quality We're looking to grow the quality rather than processing for it. I'll cover some of the more significant stuff in detail, but a lot of it I'll just touch on because there simply isn't time. Some of the research we've done in collaboration with other research agencies, and that usually means ag research, which is actually a Crown Research Institute, so it's government funded. We've done some farm-based research with cooperation from farmers who've allowed us access to their properties, answered all my questions, and basically been very good about it all. And we've also bought some animals and actually housed them ourselves or hired leased grazing for them. Just a few of the defects we've looked at. Cockle, white spot, I'll explain a bit more about those later. Two-hole and rib, which I'm sure will be familiar to anyone who's processed sheepskins. And on cattle, the major things we've looked at are an infection called dermatophilosis, scratches, which are a huge problem on hides, and looseness, and we're still actually working on the looseness. Farm fault survey is something that started in 1988. It was actually started by another researcher, but then I took it over about 6 months after it started. So we look at 13 faults on pickled pelts and 10 faults on wet blue hides, and the fellmongers and wet blue processors actually do the inspections for us, fill in our form, and send in a form monthly to say what faults, if it's 200 hides per month or 400 skins per month, have on them. And this is really useful because it allows us to determine which faults are the most common and look at trends in the fault levels and to select the most relevant faults to research, usually the most common ones. It's also very, very useful if you're applying for government funding to be able to say, well, here, look, this is what the figures say. I'll just talk a little bit about cockle, which was one of the first things I worked on. I hope those look like lumps on the skin. Sometimes they look like lumps, sometimes they look like hollows, but they're actually lumps. They're visible on the raw skin if you shave the sheep, which nobody ever does, but they're visible on pickled pelt and right through to leather and present quite a problem. They're little hard lumps, which really ruin the whole thing. A lot of work was actually done in the 1970s, and they'd already worked out that it was most prevalent in the winter. That if you shore the sheep, they either didn't get cockle or if they had it went away, and that if you treated it with an insecticide, it went away. And this just shows where it's most prevalent. This is actually from our farm fault survey. It's a 10-year average. July there is actually midwinter if you happen to be in the southern hemisphere, so that's when it's most prevalent. It goes right down in summer, which is for us January and December. And from the evidence they had, they concluded that because slower growing sheep had more cockle, that growth rates might have something to do with it. For some reason, even though insecticide cured it, they decided it wasn't caused by insects, and they decided that mould was the most likely cause. From what evidence, I'm not certain, but whatever. But from 1989, we did some more work, first in collaboration with ag research, and the basic finding of this was if the lambs were lousy, they had cockle. If they didn't have lice, they didn't have cockle. And if they were lousy and you treated them with insecticide, they didn't have cockle either. And then later alone did some research just looking at how long it took to resolve. And the interesting thing about this was that you could, after 18 weeks after treatment, there was no cockle in the pickled pelt, so you could sell them as first grade pelts. But if you then tanned them through leather, it was still visible for at least 24 weeks. So you had a 6-week period where you'd be selling in good faith pelts as first class, and getting complaints from the tanner who then tanned them because they had a defect on them. So our new conclusions were very obviously, that lice cause cockle and that prevention is better than cure because that way you wouldn't get the lingering effects on the leather. And we actually recommended a change to the standard practice of dipping in autumn. Farmers like to get value for money, and sheep are most lousy in autumn, so they dip them in autumn so that they can see that they're getting value for their dipping money. You know, a lot of lice, and look, we killed them, but we actually suggested that they do it when they didn't have many lice, just after shearing, and that would give a better result. And the end result was that the level of cockle has now gone down to less than 2%, and it's basically no longer a problem. So that was one of our successes. Just touch on these ones. White spot is a defect that you can see on wet blue as a pale spot, which is why it's called white spot. And we did some work with housed sheep and found it was actually caused by an infection called dermatophilosis. The defect pinhole we found was related to wall bulk. The breed was not so important, it was the wall bulk itself. So you could have standard Romneys, and if they'd been selected for wall bulk, which some farmers do because you get paid, or for one stage you got paid more for bulky wool, there was more pinhole than pelts. And rib is a breed-related fault. Now I'll just touch on cattle as well. Dermatophilosis in cattle, rather than white spot, causes a defect which actually looks very similar to tick, small pits in the hide. I'll put a picture of that later. In scratches, scratches was an interesting one because we spent a lot of time trying to tell farmers to remove barbed wire, go through the yards and remove nails that were sticking out, basically remove anything that cattle could scratch on. But we found that it didn't matter. Removing barbed wire wasn't the answer. If you removed the lice, which made them scratch, that they didn't have scratches, essentially. We even gave some animals barbed wire to scratch on and remove their lice, and they just looked at the barbed wire and went, oh yeah, interesting, but didn't actually use it for scratching. So for dermatophilosis, we used housed cattle, and we artificially infected them with dermatophilosis, and then at the appropriate intervals slaughtered them and processed the hides, or we didn't slaughter them, we had them slaughtered and processed the hides. And these are just, that's one of my cattle beasts standing there with his head in the feed bin, and that just shows the shaved patches that we put on the side to actually, these patches just to infect. We infect them at different stages so that when you slaughter the beast you have infections in different stages on the hide. See here it causes a sort of crusty infection, and on the wet blue it's visible as pits. With the research on scratches, we looked at the behaviour of lousy and non-lousy cattle, and I spent many hours standing out in the paddocks, which sometimes was nice and sometimes was a bit cold and wet, actually looking at them and measuring the amount of time they spent scratching. And we also did louse counts on them. And then we looked at the hide quality. And we were also lucky enough to be able to run a treatment and quarantine program on a farm which was owned by a tannery owner, so his manager had to do exactly what we said in the way of treatment and quarantine of animals to make sure that the farm was louse-free. And the results of it, that just shows some of my calves. Those are the lousy ones. And what they're actually doing there is this angle post coming down from that big fat post, and they're all got a little bit of wire sticking out from it, and they're all busy competing to see if they can scratch their skins on it. So it just goes to show no barbed wire there, but they found something to scratch on. And you can see all there that also that the time spent scratching mirrors exactly the number of lice that they have. So get rid of the lice, get rid of the scratching. And this was the results of that quarantine program and treatment program I was telling you about. Basically doubled the percentage of hides that were in the best category fit for upholstery without scratches on, in other words, and reduced the thirds to practically nothing. So it really, really works. Get rid of the lice off the farm and you improve the hide quality no end. Now I'll just talk about the work on looseness, which is still ongoing. We took hides from 800 cattle. Before they were slaughtered, we assessed them in the yards, looked at the body condition, breed. We also knew what gender they were. All the hides, 800 hides, were labelled by punch marking a number into them, and then they were checked for looseness at both wet glue and crust. And that's actually a tremendous amount of work, 800 hides. I'll just show you some of what we did. This is the assessment in the yards. I did the assessment, photographed every pinful so I could record what was going on. In case you're wondering where I'm standing, it's on that walkway, which is about a foot wide, which has a one handrail on one side, about waist height. There's an 8-foot fall onto concrete, so I was pretty jolly careful not to fall off. especially if there was a stroppy bull and a pen underneath. But I actually quite enjoyed it. It was a good way to get some fresh air and exercise early in the morning, but again, it was a fair amount of work. And we used a body condition scoring scheme for dairy cattle, because a lot of the cattle we were looking at were actually dairy cattle. And it just means you can give a number to the body condition, depending on how the bones are sticking out, basically, or not. This is our team of 3 collecting hides at the bottom of the chute where the hides come down from the slaughter floor. So it took 3 of them there to actually haul the hides out of the bin and then punch mark them with those punches you can see there. What you can't see from this photograph is that it was incredibly wet. I ran coach for a bit of an egg giveaway with water spraying around everywhere. It was very, very cold because there was a cold draft blowing through. Very noisy because there was a lot of machinery down in this basement area, and the odd forklift speeding past that they had to dodge as well. So it was a tremendous amount of work, and they'd spend all morning doing this basically, so it was a tremendous amount of work by a lot of extra staff. There's also a staff member up on the slaughter chain itself, putting tags on them so we knew which ones to collect when they came down into the hide area. And this is one of our staff members, that's Dylan, actually doing an assessment on crust. There was a template with 18 holes in it, which was put across each side of leather, and in every hole in the template, he did it, or somebody did an assessment of looseness just using the Sucre brake scale. So you can see there, it's got one there with a fair degree of looseness. This was actually done at a tannery, which is why there's a lot of people and hides and things in the background. And this is the results we got from it, and I'll just summarise it briefly. Steers have the least loose hides, bulls have the most loose hides, if you're looking at the class of animal that you're looking at. Dairy is an awful lot worse than beef as far as looseness goes. And females have more looseness than males, so we did get a lot of useful information from this, and that was a lot of work. Just talk a little bit about breed research. This is, we've looked at these 12 breeds, some of them several times. We've looked at merino skins from various places 3 times at least, and Dorpers I think 3 times as well. Some of these would only looked at once, of course. Sort of thing we would look at is the size and shape and thickness of the skins, fat content, because New Zealand sheepskins tend to have a very high fat content. Of course, you've got to remove all the fat when you process, so the lower the fat content, the better. Strength of the final leather, of course, very important. How flat they were and how much rib and pinhole they had, and whether or not they had what we call looseness, which is basically wrinkling of the leather. And also, where it was possible, we looked at suitability for wool skin use, because a lot of New Zealand skins are actually exported salted to be tanned with wool skins, often in China. I'm not really going to talk much about the findings here, except that we love Dorpers, because whether they're purebred or 3/4 bred or half bred, they produce excellent leather. So we wish all of New Zealand would actually convert to Dorpers and forget about shearing their sheep, but it seems to be a bit harder to convince the farmers. They used to shearing their sheep, so they like sheep that they can shear. And the other thing that may surprise you about Dorpers is that crossbred skins produce really nice wool skins, which sounds counterintuitive. You've got that hairy thing on the left there, that's purebred, with the shedding its hair coat and with its black head. But what you've got to think, to get this pattern, that is not actually a white sheep with a black head, that's a black sheep with a big white spot. So if you crossbreed it, you get holes in the white spot, and that's what leads to the attractive pattern that you can see on the right there. And the other thing that happens is that all the hair falls out in processing and gets combed out of the fleece, and you get left with the really nice fine undercoat of the Dorper breed, and that produces a really nice silky wool skin. Okay, farm practice research, we looked at organic farming and its effects. And we also looked at sheep farm best practice, which was basically just going onto farms and interrogating farmers as to what they did, and then looking at the quality of their skins and seeing whether they were good, bad, or otherwise. The organic farming, I didn't realise until I actually looked up the results. We spent 9 years doing it. And we had 2 conventional mobs and 2 organic mobs. The mobs actually belonged to Ag Research, and we worked in collaboration with them. 7 times a year we went out and counted louse numbers on the ewes and on the lambs. And then each year we looked at the lambs' skins after the lambs were slaughtered and assessed the quality. That's the sort of country we were working on. That's actually down at the woolshed, so that's as flat as it ever got. Quite scrubby looking land as well. That's yours truly out in the yards, busy trying to figure out which ones we actually need and which ones we don't need. And that's why I've got a bad back. Because if you've done, about 50 of those 7 times a year, it gets quite hard on the back, especially if it's ewes, because some of them are quite heavier than me. And that's actually luxurious because we were in woolshed there. A lot of this we did out in open yards, which was especially great if it was windy or raining. And what we found was, surprise, surprise, organically raised sheep have a lot of lice, and they have poor quality skins compared to conventional sheep. And we did try some remedies, but the ones available then at least didn't kill lice. We tried a lime sulphur spray, which turned the sheep a strange whitish look, and turned their grass ear tags green, and then it rained and they had green ears, but it didn't do a thing for the lice. And we tried some organic oils, which made them smell really, really nice. They took all the numbers off the plastic ear tags, so we weren't very popular about that, because they wanted to be re-tagged. Okay, let's talk a little bit about this. Farm practice research, that involved a lot of on-farm visits, so I saw a lot of the countryside. It was done back in the days pre-GPS, so I had to follow the farmer's instructions. Farmers had no idea what colour their house is. They'd say it was blue with a red roof and you can't miss it. And when I got there, it would actually be white with a green roof. It used to be blue with a red roof, but that was back in 1963 when they actually last looked at it. So it's never been painted. And they'd always say, without fail, you can't miss it. And I'd think, yes I can. And the other thing was that they tend to plant shelter belts, which have grown since they last looked for their house. So again, usually the house was hidden behind a shelter belt. But I always found it in the end, but sometimes it took a long time. They would also say things like it's 20 minutes from town, and you go, right, okay, 40 minutes from town. Because they all drive at the speed of light on the wrong side of the road without indicating. And it does take them 20 minutes, but if somebody's actually driving legally and trying to avoid the farmers, it takes at least twice that long. But we got some really useful data out of this, because some farms produce really good pelts and some produce really awful pelts. And I interrogated them as to what their practices and treatments of the sheep were and whatever. We got some really good data. So it was really useful. I think that the best instructions I ever got for listening to a farm was go to the top of the ranges and take the third road on the left. And that was, great. I'd driven for 2 hours, and is this the top of the ranges? Is this the top of the ranges? Because the ranges were a sort of small mountain. I did find it in the end. It was the 4th road on the left, actually, by the way, but it took me a bit. Okay, now I'm just going to go on to the genetic research we've done. We've looked at deer skin quality and snip chips, which I promised you right at the very beginning, and this is currently ongoing. The skin quality of deer was looked on as part of something called the deer progeny test, which was an industry-led central progeny test, and it was with the aim of producing or improving venison production. So once again, skins were tagged on at the end of this, but it was still very interesting to be involved. There are about 900,000 to 1 million, depending on the year, farm deer in New Zealand, and I think it's the largest farm deer industry in the world. But correct me on that if I'm wrong. And this produces about 350 to 400,000 skins per year, so it is quite a big part of our industry. The test was funded by Deer Research, which is just an aggregation of farmers, Alliance, which is a meat processing plant, Landcorp, which is a very large group of government-owned farms, and again, Ag Research, the government-funded Ground Research Institute. And we looked at 310 deer skins as part of this trial. And we were trying to work out what components of deer skin quality might be genetically determined, and also whether selecting for better venison production might impact skin quality, either positively or negatively. And we found that tear strength had high heritability, and this was not only interesting from the point of view of the deer skins, but also gave us an indication that it might be heritable in other species. And there was also another feature which was moderately heritable. And we get to snip chips. It's going to give you a wee lesson on DNA. Some of you probably know more about it than me, but it's just a little bit so you know the background. Everybody knows that DNA is a double helix. They found that out just down the road from here. And held together by base pairs with 4 types of nucleotide. And you've got either adenine and thymine, which bond together, or cytosine and guanine, which bond together, and they can only bond in these pairs to hold these 2 parts of the double helix together. And if you want to replicate it, you take the strands apart and then they form a new strand using the old strand as a template, and you end up with 2 double-stranded identical strands. That's the theory anyway, if everything goes according to plan. Human DNA is about 3 billion bases long, which is about 2 meters long, which doesn't sound all that long until you realise it's got to fit inside a cell, which is not very big. So it gets coiled up into chromosomes, which are in the cell nucleus. And genes make up part of this DNA, and they're the basic unit of heredity, which most people know, but they only make up somewhere between 1 and 10% of the DNA. And they vary quite a lot in size, can be a few hundred nucleotides long, or up to 1 million or more. It's the genes which code for the proteins which basically keep life going, and the rest of the DNA, the other 90 to 99%, regulates when these proteins are made, how they're made, and how much is made, but they don't actually determine its structure. And because basically every living thing has to operate in more or less the same way with respiration and whatever, a lot of DNA is held in common. So we share 98% with chimps, which is bad enough, but 65% with hens is even worse. It explains people I know. But right, we did get to the SNPs now, which is a single nucleotide polymorphism, which is an incredibly complicated way of saying that one of those base pairs or one nucleotide base is substituted for another. And what happens when you're getting this replication with the strands coming apart and replicating, occasionally things go wrong and you end up with bases changing. So you can see there that that's a SNP. The adenine-thymine pair has been swapped for a cytosine-guanine pair. And the effect is, depending on where it is, it can be different. So if it prevents the synthesis of a useful protein, that SNP doesn't get passed on because the animal dies, basically, the organism dies. Or it can cause a change to the protein, so you end up with brown eyes instead of blue eyes, or a double muscling instead of single muscling, something like that. Or it can have absolutely no visible effect whatsoever, and most of these SNPs actually have no visible effect. An interesting thing about them, though, is they pass from generation to generation even though they have no effect. So you can use them to track ancestry. The more SNPs that 2 organisms have in common, the closer the relationship. And this is very important from our point of view because it means you can trace whether animals are related or not, just using the SNPs. And they can also be used as gene markers, so they might not actually determine any observable trait that it's got, but they can be used as a marker for a gene, and that's also very important. And it just happens if it's very close to it, but not actually part of the gene. And it's actually possible to identify in sheep, you can identify 600,000 of these SNPs. So how does it work? And this is, I hope nobody here knows how SNPs work, because it's a very, very simplified version of it, and probably not strictly accurate. But you take several thousand sheep, and you measure whatever it is that you want to measure, whether it be lamb survivability or carcass weight or growth rate or whatever. And then you identify all the SNPs you can find of all of the thousands of sheep. and then you correlate which SNPs go with which variation in the trait. And then once you've done that, you know which SNPs to target, so you can develop a specific SNP chip to actually target those particular traits. And how does the chip work? Millions of glass beads with a bit of SNP detector strand attached to each one obviously. And you take the DNA apart, so you've got single strand of DNA, mix the two together, and the test DNA then bonds to the detector strand on the glass bead, and you use a technique to make it scannable. I have no idea what the technique is, so please don't ask me. And this is what the SNP chip actually looks like. And these are now commercially available. There's a trick to determine parentage, the cheapest and simplest version of it. It's known for obvious reasons as Shepherd. But you can also determine 22 traits ranging from carcass weight to facial eczema resistance in sheep by using these larger and obviously more expensive chips. And it's used quite often by stud breeders, like they'll test their lambs and then they'll be able to tell potential purchasers of those lambs for breeding purposes whether they carry a gene for facial eczema resistance or what sort of growth rates they can expect from the lambs, that sort of thing. And the advantages are, for a start, you don't have to identify parentage of stud animals at birth. You can do it later, which is really useful if you're not chasing animals around in the rain and trying to catch them and read their ear tags. You can also identify traits in the while the animal is still alive, so you can use it for breeding. I mean, it's absolutely no use whatsoever to determine that a particular lamb has got a really good growth rate if the only way you find out is after you've slaughtered it. And you could use test breeding, of course, to do this, but using SNPs is a lot faster. You don't have to wait a generation. It's also cheap. The Borula gene is a gene which is responsible for multiple births, and they actually found this back in the 80s, and it cost them $5 million to find it. Now you can do that with SNPs, and it costs you $5,000, so it's just a huge difference. And this is a summary of it. Basically, you can identify the parentage of these little guys, and if you're using a more advanced chip, you can also work out who's going to be used for breeding and who might have a different future. And what we've done to date, we've taken part in an ongoing Farm IQ primary growth partnership study, and that's just basically a group coming together to look at this. And so far we've assessed skins from 300 lambs, and it was thanks to the partners in Farm IQ that we were able to be involved in this. Landcorp again, a common farming agency. Silverfern Farms was the processor involved in this case, and once again, Ag Research. We identified the skins using the SNP chip technology, so we could tell who was related to whom, and who their sires were, who their mothers were. And this means that you can actually determine heritability, which is a huge thing to be able to do. Normally, if you were doing this sort of trial, you'd only be able to do a very few animals, and to identify the skins is really difficult. The best we can ever do is stand up on the slaughter chain, which is look at the number, tag number of the lamb, and then have somebody further down the chain tagging the skins with consecutive numbers. And if all goes well, and nobody drops a tag or puts a tag in the wrong skin, you can maybe go up to about 50 skins, and then it all starts to fall apart. Whereas with this, we did 300 quite easily. Well, I say quite easily, I was involved in tagging somebody else had to do all that hard work. But then we were able to carry out a lot of measurements and assessments on the skins. And we found heritability in some fairly important traits, including tensile strength and grain strength and severity of grain strain. So this was actually a hugely important finding. This is just to show you the various bits of equipment used. That tissue sampling unit here, that's about 1 inch long in total. That's a little punch. That's the actual tube the sample goes into, and that full of the ears there is just something to hold it by. This applicator looks exactly like an ear tagging gun, because usually the samples are taken from ears rather than from skins. And that's just a loaded applicator, the other one. There's one of our people actually taking the sample with the little punch there is about to go through and push the sample into the tube on the other side. can see here, if you look really, really hard, there's a sample sitting just in there, ready to be sent off to for its analysis. Currently, we are working on the extension of the work. We did 300 skins, with another 500 skins. We've actually got the skins now. We've taken the samples, they've been sent off for analysis, and we're in the process of having the skins processed. And having 800 rather than 300 means that we can get much better figures on heritability, and maybe able to see heritabilities that weren't visible with only 300 skins. So it's really going to strengthen our data quite a lot. And the other thing we're looking at on a separate study is determining whether genetic markers exist for the sort of properties which change with age, and especially strength. So we're looking at milk lambs, older lambs, and ancient ewes, and we've also, because we really, really like them, we've included Dorkers in this study. If I remember correctly, thank you very much. That's the end of my talk. I'd just like to acknowledge the people. Much of this work has actually been funded by what is now called the Ministry of Business, Innovation and Employment. It used to be called the Foundation for Research, Science and Technology, and then it turned into the Ministry for Science and Innovation before finally becoming MBIE. But they're all the same ministry basically. It's what ministries do. When they've got anything better to do, they change their name and get some new stationery. Ag Research and also Massey University have at various stages been involved either as collaborators or as subcontractors in some of the work. I have to thank the farmers in spite of their terrible instructions on how to get there. They've been really, really helpful to me, and they've been really generous with their morning and afternoon teas and lunches as well. And many of the LASRA members who have provided access to their processing facilities, and also we have something called the ITAG, which is the Industrial Technical Advisory Group, to which we present all our research, and they tell us whether they like it or not, and also tell us what they would like researched, so that they've been incredibly helpful over the years. And of course all the LASRA staff who help them, whether they were mouse counting or recording data or helping clean out the animal crates or chasing the sheep around the yards. People are actually incredibly useless as sheepdogs. We'd have sheep standing there going, "Gate, what gate? I see no gate." But while we jumped up and down and waved things and screamed and shouted, and along would come a big New Zealand hunterway, which are the barking sheepdogs, and go woof, and the sheep would go, "Gate, I see the gate," and run into it and meet queues and line up to be drafted or whatever we wanted. But anyway, the LASRA staff did try. So thank you very much.